Hi, everyone. I'm Eric, a PhD student in biomedical engineering at Georgia Tech and Emory University. Thanks a lot for dropping by my presentation. In brain stimulation, whether it's optogenetic or electrical, we usually select stimulation parameters by simple and arbitrary conventions. That might include constant frequency and constant amplitude waveforms, and we might have loose hypotheses like if we know we have a biomarker at X frequency, then we'll want to modulate at that frequency. But more recent work is exploring novel, complex, and irregular types of stimulation waveforms that can produce new and different types of changes in neural activity and provide therapeutic benefit in a bunch of different applications. Now, there are two key barriers to understanding this world. There's really an infinite number of different ways that we can stimulate the brain, and we don't know what any of those parameters are going to do until we empirically try them out, which becomes an intractable problem. In this study, we're taking a computational approach to do two different things. We're going to understand the effects of different stimulation parameters on brain activity. And two, we're going to design an engineering approach using machine learning to select optimal stimulation parameters to control brain activity based on feedback. For our methods, we're stimulating in the medial septum of rats, which is a brain region known to be a key pacemaker of oscillations in the hippocampus. We're stimulating with channel rhodopsin 2, which is excitatory stimulation, and we're using the pan-neuronal HSIN promoter. And meanwhile, we're recording simultaneously from 16 channels across CA1 and CA3 of the hippocampus. We comprehensively evaluated 5 different stimulation parameter spaces. First is the standard pulse train space, which is defined by the amplitude, frequency, and width of constant frequency pulses. Second is the sinusoid space, which is defined by an amplitude and a frequency. Third is the Poisson pulse space, which is defined by the amplitude, frequency, and width of pulses, just like the standard pulse space, except now the interval between pulses is randomized and drawn from a Poisson distribution at the given frequency. There's the nested pulse train space, which has 2 new stimulation parameters on top of the standard pulse space. There's the width and frequency of trains of pulses, and then there's also the width and frequency of the pulses within each train. Last is the double sinusoid space, which is the combination of 2 different frequency sinusoids. First, we asked what are the effects of these different stimulation parameters on 2 variables of interest in the hippocampus, which are gamma power and theta power. We found that the effects of gamma power are straightforward and pretty consistent across the different stimulation parameter spaces. As you increase the amplitude and the frequency to about 40 hertz or so, you get a strong and robust increase in gamma power, and that was pretty consistent across the parameter spaces. Modulating theta power is a lot less straightforward. We actually found that none of these different stimulation parameter spaces was actually able to increase theta power beyond baseline. This is especially interesting with the nested pulse train space because several prior studies in the literature had specifically selected this space based on the idea of entraining low frequency oscillations. So we were surprised at this result. When looking at the standard pulse train space, we found that the optimal stimulation parameters that increased theta power were invariant with frequency and were actually low as opposed to high amplitude, implying that the optimal way to increase this biomarker was to not stimulate at all. We further investigated this effect by looking at the change in power at stimulation frequency across the full range of low to high stimulation frequency. And first, we confirmed this effect on low frequency as a function of these different stimulation parameter spaces. And we found that the ability to entrain oscillations really started to increase past 10 to 15 hertz or so, and further increased past 30 hertz. We also looked at this effect under anesthesia, and we found that the ability to entrain high frequency oscillations was suppressed under anesthesia past 25 hertz or so, relative to awake behavior. Looking at the Poisson pulse space, we found that the effects of different stimulation frequency parameters on gamma power was broadened, and that's because the Poisson pulse stimulation produces broadband excitation in the power spectrum. Last, we compared how these different stimulation parameter spaces produce different changes in multi-band activity by using the UMAP dimensionality reduction algorithm to take all of the different power spectral density points produced by these different stimulation parameter spaces and learning a single manifold representation to compare all of them. And then we colorize the different stimulation parameter spaces on top of this manifold. We also compared the stimulation parameter induced activity to behavior recorded during natural activity, which is shown in black. This was during 2 different behaviors, which were exploratory behavior and quiescent behavior. And we are starting to see a few different interesting qualitative observations. First is that the standard pulse space in light green produces activity patterns that are most different from natural activity. Then if you look at the light blue and red points, which are the double sinusoid and nested pulse train space, we see that these two larger stimulation parameter spaces have a greater representation in the neural activity space, implying that they can produce more diverse changes of neural activity with respect to multiple bands. And they actually have the greatest overlap with the black points being the points recorded during natural activity, meaning that these multi-band, these more complex stimulation parameter spaces are essential to producing activity that more closely resembles natural activity. Next steps for this analysis are going to be for us to dive into the different areas of this parameter space and start to see what exactly are the new parameters and the activity that correspond to these different unique regions of the dimensionality reduction space. In parallel, we're starting to work on developing an approach for optimizing stimulation parameters. So we view the brain as a black box, and we can try out a set of stimulation parameters, and then we can measure an objective function. That'll be the similarity or the difference between induced and desired neural activity. And then we can use a data-driven search strategy such as Bayesian optimization to cycle back and forth and iterate towards the stimulation parameters that optimize that objective function. And this works kind of like this. We can fit a regression model to a few different tested stimulation parameters, and then slowly select stimulation parameters that iterate towards those that optimize our objective function. In conclusion, entraining low-frequency stimulation activity is challenging, and that appears to be resilient to different stimulation parameter spaces. Conventional stimulation appears to induce artificial changes of activity, and new and more complex stimulation parameters may be able to produce more diverse and within manifold activity patterns that are closer to the spectrum of natural behavior. So of course, I'll have to thank my advisors and my lab that made this possible, and I'm happy to take any questions. Thank you very much.